Welcome to the Sand Science Experience podcast, where we explore the latest and most exciting papers and preprints in science.
If you enjoy what we do, please take a moment to like, subscribe, and maybe share this with friends and family. It really does support the Sand Science Experience.
So today we are tackling a really persistent paradox in the biology of aging. It's all about the mitochondria.
Right, the powerhouse of the cell. And it's confusing, isn't it? Because for years, you see these studies with completely opposite results.
Exactly. One paper will show that damaging mitochondria shortens life, which, makes total sense.
Of course, less energy, more problems.
But then another study comes out where, a mild impairment actually makes the organism live longer. How can both be true?
Well, this new paper we're looking at today suggests the answer isn't what is happening to the mitochondria, but when. The timing seems to be everything.
And the researchers used the common fruit fly, Drosophila, to explore this idea of an evolutionary trade-off.
Yeah, the thinking is that for a short-lived species like a fly, the number one priority is immediate reproduction. So it might sacrifice, say, long-term body maintenance to make that happen.
And a core part of that maintenance is the energy system itself, the process called oxidative phosphorylation, or OXPHOS.
This whole paper really builds towards one core novel idea, which we'll get to. But to understand it, we first need to see how they set up the experiment.
Right. They created a model of aged mitochondria by disrupting a specific part of the chain called complex 4.
And that's a key choice. Disrupting complex 4 doesn't just cut energy production. It also, and this is crucial, it increases reactive oxygen species, or ROS.
That oxidative stress, that's such a hallmark of aging.
Precisely. So let's quickly go over the background results that set the stage for the big reveal. First, they looked at when they applied this damage.
And they found two completely different outcomes, right?
Totally different. If they induced the complex 4 knockdown during development, while the fly was still a larva, the adults were, well, they were a mess.
How bad are we talking?
Catastrophically short-lived. Their lifespans were cut by like 60, sometimes almost 70%.
Wow. Okay, so that's a huge effect. What about when they did it only in the adults?
Much, much milder. The same genetic damage, but applied only in adulthood, led to maybe a 10 or 20% reduction in lifespan. Still bad, but nowhere near the same systemic failure.
So the damage done early is what really causes the problem. And that problem was, what, a kind of metabolic chaos?
It was. The flies that had this developmental damage were basically in a state of chronic starvation as adults, even with plenty of food.
Their fat body, which is like our liver and fat tissue combined, couldn't store energy properly.
Exactly. They had no reserves. And their cellular recycling system, Autophagy, was completely overwhelmed. You could see it with the buildup of this protein, Ref2P. It's a clear sign that the whole system is clogged up and failing.
So if the damage is done during development, can you also fix it during development?
That's the next logical step they took. And the answer was a resounding yes. They tried 2 different rescue strategies, but only applied them during that developmental window.
The first one was pretty clever. They didn't fix complex 4, they just built a bypass around it.
Yeah, using a gene from a sea squirt called alternative oxidase, or AOX. It just gives the electrons another route to go, relieving the pressure. And it worked beautifully. Lifespan was extended by over 60%.
And the other intervention, that one targeted a downstream pathway, right?
Right. They activated the TO pathway, which basically tells the cell, hey, resources are abundant, store some fat. And that also worked. It rescued lifespan by about the same amount.
So, to sum up, damage during development is catastrophic. But interventions during development can completely fix it.
Exactly. And all of that is the essential setup for the most novel part of this study, which is what they found in Figure 4.
This is where it gets really interesting. Because the obvious next question is, can you fix the damage later in life?
The researchers tried to do just that. They took flies with a developmental damage, and then, in adulthood, they switched on a gene to produce a fresh working copy of the broken part of complex 4.
They gave the cell the blueprint and all the parts to make the repair.
They did. And it completely failed.
Failed.
Completely. It did nothing. Turning on the rescue gene in the adult fly did not restore mitochondrial respiration at all. The damage was locked in.
Oh, wait, so why? Why couldn't the cell just replace the broken part?
Well, this is the core finding. It seems the cell just doesn't. When they looked at protein turnover, they found something remarkable.
Oh.
The instructions for building these OXPHOS components, the mRNA, Their levels drop right after the fly becomes an adult,
Mhm.
but the actual proteins themselves, they just
They
stay.
stay for how long?
For the entire adult life of the fly,
Yeah.
these huge protein complexes are incredibly stable. They're built once during development, and then they're essentially never replaced.
So the cell builds its power grid once, and that's the grid it has for life. There's no maintenance crew.
That's a perfect analogy. The maintenance crew gets laid off right after construction is finished. These OXPHOS proteins have some of the longest lifespans of any proteins in the cell.
And they proved this wasn't just a fluke with complex 4, right? They tried it with complex 1 as well.
They did. They tried to knock down a complex 1 subunit in adult flies. They hit it with RNAi for 28 straight days. Nothing. No effect on respiration. Once that developmental window closed, the mitochondrial respiratory capacity was fixed. It could not be significantly changed, for better or for worse.
So this really provides an evolutionary explanation for that paradox we started with.
It does. For a short-lived animal like a fly, the strategy is build a high-capacity energy system once, go all out on reproduction, and don't worry about long-term maintenance.
The cost of that strategy is a complete loss of metabolic flexibility in adulthood.
Right. And it means that aging in a fly isn't just a sped-up version of aging in a human. It's following a completely different rule book, one that's written around reproductive urgency.
The fact that this fundamental energy system is locked in so early in life leaves us with a really provocative thought to consider. If a fly's metabolic fate is sealed in its first few days, how much of our own long-term metabolic health, our ability to handle stress for decades, is actually determined by cues we receive during our own development, long before we were even adults?
It definitely suggests that the most critical window for intervention might be much, much earlier than we've been looking. It really shifts the perspective.
A fascinating shift. We hope you enjoyed this deep dive. As always, please remember to like and subscribe.
Your support truly helps us continue to bring you this content from the Sand Science